Hello everyone, I am Aditya and welcome to CCMB Conversations. In CCMB Conversations, we will take you to understanding work in science that is happening around us through experts. Today we have with us Dr. Kevin Gardner, the Director of the Structural Biology Initiative at the CUNY Advanced Science Research Center, USA. Welcome, Kevin.
Thank you. Glad to be here.
Can you explain what your work is?
Gladly. So consider me a biochemist who, like many people in the biological sciences, is interested in a really important question. How do cells know what's going on in the world around them? Whether they're single bacteria or yeast living out in the environment looking for nutrients, or signals of our cells within our own bodies responding to signals from other cells, that kind of detection as to what's going on around a cell, being able to measure, say, levels of a chemical, and then determine whether that's above or below a certain threshold and get the cell to react accordingly, is really fundamental to life, right? Cells want to be attracted to nutrients and want to be move away from repellents or produce different protection mechanisms for that. I believe very firmly that through a lot of biochemistry and a lot of structural biology, we can look at the machines that do that switching, understand how they work normally in biology, how they can be broken in disease, and how we might learn some powerful lessons from them to develop new tools for biotechnology or new therapies for human disease.
The first thing I would like to ask is when we talk about using these for therapy in human disease, what kind of potential are we looking at?
Right. So really critical for the kinds of proteins I'm talking about, ones that sense changes around a cell, transmit that information inside, and then inside the cell and let the cell react. When we start talking about therapeutic human disease, the kings of the hill here are the G protein coupled receptors, the GPCRs. Those are the target of, depending on who's doing the statistics, 40 or 50% of all known pharmacologic compounds that are being used are being directed at GPCRs to control intracellular biology. However, if we look, the same principles, proteins that have evolved to sense different stimuli and respond to them, come in many different kinds of flavors, other than just the GPCRs. So we've looked at a kind of such switch called the PAS domain. It is conserved and used very widely to do such sensing, not only in us, especially in parts of our hypoxic response, parts of our response to foreign compounds, or xenobiotics, also parts of our circadian clock, a system which is being somewhat challenged inside of me as I arrived here from New York about a day ago. Those are the targets that are quite clear that I think if we learn how they can be switched on and off, where we can have great therapeutic benefit. I'll note that that is an area of interest in to many groups, but I'm really proud that group that my lab was part of was the first to successfully demonstrate this over in the hypoxia sensing system, which our cells use, our bodies use, to respond to low O2 and turn on a variety of ways to help cells adapt to low oxygen. That's great when you do it transiently. For example, if we go from here in Hyderabad up to the top of the Himalayas, you would turn the system on and it would protect your body from many of the physiologic challenges of that. However, when that system's turned on all the time, you are unfortunately unleashing a variety of growth factors and other signals in your body that are very highly predisposed towards cancer formation. Individuals who have a genetically inherited disease, von Hippel-Lindau syndrome, that turns one of these systems on, have literally a 70% chance of developing cancer in their lifetime. Taking advantage of that knowledge from human genetics and partnering it with the kinds of structural biology and biochemistry we do, that allowed us to go through, know what proteins we should solve high-resolution structures of, figure out what kinds of small molecules might bind them, and work together with chemists to make those initial compounds that were only good for working in a dish in the lab and turn them into something that started working fantastically in cells, and then in animal models, and then in clinical trials. That's now a drug in the United States and about 19 other countries, I believe, that is used as a way to shut this system down at times of our choosing, even in the individuals who have it turned on genetically all the time. Roughly about 10,000 people in the U.S. are taking that daily as a result.
So you've designed a compound which turns off hypoxia-inducible factor 2?
Precisely.
Okay.
Yeah, and that, if there are chemists reading your article or listening to this podcast, I can imagine some of them shouting at the article or at the podcast because this is a traditionally quote-unquote undruggable class of targets. What are we really good at designing drugs for in 2026? We're really good at designing them towards things like GPCRs or enzymes. Those have clearly definable sites and activities that have been easy to target. When you start hearing about transcription factors being turned off, well, that starts getting challenging because these are now not only just intracellular, but intranuclear targets. They don't have an enzymatic activity per se. And indeed, what you're trying to hurt are protein-protein interactions between, in this case for HIF, two different proteins that must assemble to form the active HIF2 transcription factor. We were, in some degrees, lucky that we found a novel mechanism in HIF2 that made it especially druggable, even though it is in an undruggable class. But I'd also say that was guided a bit by intuition and a good guess. Because the same kind of sensory module that is being used in HIF2, for example, which senses oxygen through some very elaborate mechanisms. I encourage your readers or listeners to go into the 2019 Nobel Prize in Physiology and Medicine, see very elegant set of post-translational modifications there in particular. But it turns out that this had retained an evolutionary footprint or remnant, if you will, of much simpler oxygen sensing and light sensing systems from the plants I'm looking at outside of our window or bacteria growing in the environment, where here they would directly bind things like heme or riboflavin or FMN, all of which can be redox sensitive to SENSO2, but do so very deliberately inside of the domain that we see now echoed in the HIF2 transcription factor. So plants and bacteria, I think, give us very clear inspiration. Something should bind in the middle of this protein and regulate its activity. We don't know what the natural factors are that do that in those simpler systems. Or in HIF2, we built a drug that does it at a time of our choosing.
So you've also mentioned, or maybe I have read about it, about light-sensitive proteins.
Right,
you can turn them on and off using light.
Yeah, very much so. And I know you mentioned in our discussions before, you were talking about your background being in neurobiology. The field of neuroscience has been had a wonderful set of tools delivered to them by the identification of light-controlled ion channels, known as channel rhodopsin. More generally, kind of technology is called optogenetics. Let's take genetically encoded photosensors from biology and repurpose them to go do different things. Well, as part of our interest in understanding how environmental signals turn proteins on and off, we did a fairly deep dive in the field to start looking for novel light-regulated proteins. Actually, people who come by my lab in New York, you'll see that the lab has two subgroups in it, one called the blue crew, looking at blue light sensing, and another one called the O2 crew, looking at oxygen sensing systems. And for the blue crew, we know in this family of past domains, the subset of them are sensitive to blue light illumination, initially identified by really transformative work by Winslow Briggs and coworkers at Carnegie Institution of Science, the Stanford campus, where they went on a decade-long journey asking how plants know how to respond to the day-night cycle, obviously critical for their life. And that revealed to them, for example, some light-regulated serine kinases. And structural work by my group showed a key part of that switch, how a certain part of a protein can be well folded in the dark and then very unfolded in the light. And we think we understand how that controls kinase activity. But then the photobiology drives so much more than just plants watching the day-night cycle. So as we look around in biology, we ask, where else has evolution delivered us new light control tools? It's light control transcription factors and other kinases and phosphatases and parts of the ubiquitination machinery. So the one that's received, I think, the most utility, not just as a structural biology or biophysics tool for groups like mine, but for cancer biologists, for neuroscientists, for cell biologists, has been a protein from marine bacteria, where people may or may not be aware that light goes into the water, especially in a marine environment, and penetrates to very different depths depending on wavelength. Blue goes the deepest, up to 100 meters in a relatively clear environment. And as a result, it is a good signal to organisms which may be facultative phototrophs. Ones that'll be photosynthetic by day and autotrophic, eat whatever they can find by night. Blue light is not just a watch telling them what time of day it is, but it's also a depth gauge as to where they are in the water column. And so some marine bacteria turn out to carry some very interesting little blue light photosensor proteins, including one that's a DNA binding protein. It will only bind DNA in the light and not in the dark. So my group did a lot of hard work in biochemistry and structural biology to figure out how this switch worked. Very elegant amount of protein conformational changes occurring. But then we engineered it to learn a few important things like what a nucleus is and what RNA polymerase II in eukaryotes are. So now it's a widely used tool by groups doing everything from yeast genetics to developmental biology. Actually, you mentioned your background includes zebrafish. We've been working with zebrafish labs using it. So now you can go into, say, a developing zebrafish embryo at a specific point, light up a specific set of cells, and turn on a long-lived fluorescent reporter, say to be able to mark a lineage, or turn on potent cell toxins to ablate a subset of cells to do an experiment to ask what happens if not just all cells, not just a certain type, but these exact cells we're painting with light, what if they're not there any longer? It's really been a fantastically fun part of our work to go see not just what happens within the four walls of my lab, but more broadly in biology with a new tool, a new idea. One philosophical comment, I mean, we're all here as scientists standing on the shoulders of giants who came before us. The experiments that worked and the experiments that didn't taught us the foundation that we work with now. I think one of the most flattering things that I see in work that from my group that gets used widely elsewhere, or I hope the groups whose work we're using understand how we're complementing them. If something is useful, that enables the next set of questions. What could be more powerful than that?
That's fantastic. I have one very naive question.
Please.
So when we talk about ligands being delivered to the hypoxia factors,
right,
or when we talk about optogenetics, how do we make sure that it is targeted, it is reaching the right spot? Because we cannot keep activating everything.
Right. So the easiest one is, let's start with light, and blue in particular. So there are multiple ways that you can selectively apply this. Nicely within us, as higher metazoans, as humans, right? The bulk of response to blue light is coming through our eyes and through the rhodopsin system. Elsewhere in our body, it turns out we've not evolved to have a lot of these blue light photoswitches in. So we can use a combination of good genetics, for example, delivering, say, through viral delivery systems, ways to express the photosensor in a tissue or cell line specific manner. So the photosensor never even gets turned on in the cells that you don't want it to actually have some control over. And then a challenge with blue light is it doesn't penetrate well. I encourage listeners to go find their nearest red laser pointer at your finger. You'll see it'll light your entire finger up. Blue light laser pointers are a fair bit harder to find, but should you find one and illuminate your finger, you'll see it barely goes anywhere at all. We can use that to our advantage here, though. And if we have trying to illuminate a portion within a mouse, within a person, then you deliver, you can get hardware systems with, for example, some very elegant, very fine fiber optics to deliver light into only specific regions. So that combination, genetics, so you only have the receptor at one spot, hardware only deliver the blue light into a certain spot, it's exquisitely sensitive. Over for small molecules, this is a challenge that pharmacology has been dealing with for over 100 years. So we rely here on medicinal chemists who are good at tuning the delivery properties of the small molecule to be able to go into certain tissues and not into others. But here it is really systemic exposure that we're going for. especially for a cancer therapy, is a good thing, right? We're trying to go after a primary tumor at one spot. And unfortunately, if the cancer is caught relatively late, we could be dealing with metastases throughout one's body, and you'd like to be able to have broad general exposure. Here we take advantage of the fact that we learn from biology. Where is this system turned on, and to what degree? And know that we will tailor our degree of inhibition so we kind of target the most beneficial effects, stopping the cancers, without going to doses that incur off-target effects. Now, I need to be clear, I'm a biochemist and structural biologist. I understand those things in principle and in theory. I don't do clinical trials on my own. That's not my area of training in the least. But it's been wonderful to work with individuals who have those experiences in medicinal chemistry, in clinical trials, and watch them work. I'm very curious about it, and just as they're curious to understand kind of how we got those molecules coming in their direction in the first place.
I think I got everything that I wanted. Thank you.
Wonderful. Thank you so much.
Thank you so much. Do you have anything like a final take-home message for the listeners?
I think I want to draw on that last point that I mentioned about the importance of interdisciplinarity. It is something that I found absolutely critical to my work in the regard that I think there are things we try doing in the lab. I know what my group and I are really good at: in vitro biochemistry, structural biology, and beginning to move things into simple cellular systems. But if we're serious about this, we need chemists, I need hardcore cell biologists and cancer biologists, and people who then can go on and do some interesting engineering or pharmacology application work. But I think from my personal perspective, that kind of interdisciplinary work is really driven by several things in the individuals. We each need to be experts in our own disciplines, number one. Number two, we need to be good listeners. We're going to meet other good disciplinarians, and we need to learn what we can from them and their work and how we can work well together. And then number three, be curious enough to want to engage in conversations. And in ways, we're all smart people. We're all doing a lot of hard work in science, and we can easily take a conversation into nomenclature and jargon that is understood only by the experts of the experts. That can be important in some settings, but especially one where you want your work to be able to go more broadly. You need to maintain that rigor and being able to speak with your technical expert colleagues, but you need to be a little more curious, be a good listener, and find good ways to communicate with others if you want to have your work achieve more broad impact. That's what I've got.
Thank you so much.
Thank you so much. It's been a real pleasure.